Hey everybody, welcome to the next lecture on YouTube. Today we're going to be talking about cell structure and function. Most of this is review for many of you. I know those of you who had me in 7th grade, we did this extensively, and I'm sure those of you who did not have me probably covered this at some point in the last couple years. So we're going to go through it kind of quickly, and hopefully you will be able to pick up a lot of this stuff as we go along. All right, so first thing we're going to talk about is exactly how cells were discovered and kind of how we classified cells. So first guy that discovered cells was Robert Hooke in 1665. He used a set of magnifying glasses to make a primitive microscope, and he was able to see cells using that microscope. Some early studies of cells were conducted by Matthias Schleiden and Theodor Schwann, and those two scientists proposed what is now known as the cell theory. So you guys need to know all these scientists for sure, and also you need to know what the cell theory is. So the first part of cell theory states that all organisms are composed of cells, that cells are the smallest living things on Earth, and that cells arise only from pre-existing cells. So those three things definitely need to know going to the test. All cells today represent a continuous line of descent from the first living cells. So knowing these three qualifiers for the cell theory, very important. So continue with the cell theory, some other things that all cells have in common. So these are all cells, including prokaryotic cells and eukaryotic cells. So all cells have genetic material. All cells have what is called a cytoplasm, which is this semi-fluid kind of space. We call it a matrix. So all the space that's filled up by this fluid inside the cell is called the cytoplasm. And all cells have plasma membrane, which again is composed of phospholipid bilayer. Prokaryotic cells. So we're going to classify prokaryotic cells real quick and then talk about eukaryotic cells, and then we'll go into the structure and function of all the organelles. So prokaryotic cells, one of the biggest things about to identify prokaryotic cell is they lack any kind of nucleus. Their genetic material is present, but it's not in a nucleus. It's called a nucleoid. I'll show you a picture of that. So again, they do have DNA, but it's just not in the nucleus. Two types of prokaryotes. There are archaea, which is this really primitive type of organism that only lives in really extreme environments like volcanic vents and like Salt Lake, like the Salt Lake, really extreme environments. And then bacteria, of course. Prokaryotic cells, they possess genetic material in the nucleoid, like I already stated. They do have a cytoplasm. They do have a plasma membrane. They actually have a cell wall, which is made out of this protein sugar called peptidoglycan. And then again, prokaryotes have ribosomes. That's usually a common question on tests. What that I ask a lot of the time is, which of the following do prokaryotes not have? And a lot of people confuse ribosomes. They do have ribosomes, so make sure to make a note of that. Prokaryotes do have ribosomes. They do need to make proteins. And then, of course, they don't have any membrane-bound organelles. So organelles like the Golgi apparatus, like the endoplasmic reticulum, those organelles, they don't have any of those. And the reason we call them membrane-bound is because they're held by the plasma membrane. And they're also held within a membrane themselves. So here's a picture of a prokaryotic cell. This is a flagellum. They use that for locomotion, to move. You have pili, which are sometimes used as a way of exchanging genetic information. But the main thing I want you to be able to identify in this picture is that, again, here is the nucleoid, this is the DNA, and this huge space right here is considered the nuclei, or nucleoid. And then they also have ribosomes, they have a cytoplasm, they have a plasma membrane, and they have a cell wall. Okay, so these are the most important things to know, these four right here. Prokaryotic cell walls are designed to protect the cell and maintain the cell shape. The flagella is present in some prokaryotic cells, and again, it's used for locomotion. And again, it uses this rotary motion, so rotary spins, and that kind of does a whip, causes the flagella to whip, and that propels the cell. You can kind of see that here. All right, eukaryotic cells. Eukaryotic cells do possess a nucleus, so all eukaryotic cells have a nucleus. They are much more complex than prokaryotic cells. Prokaryotic cells are actually very simple. They're also much, much smaller than eukaryotic cells. And they're able to compartmentalize many cellular functions within what are called organelles and what is called the endomembrane system, which we'll discuss. And then eukaryotic cells possess what is called a cytoskeleton, which is used for support and maintain the cell structure. So here's kind of a broad picture of the eukaryotic cell. You can kind of see the nucleus, the cytoskeleton, which is right here. You see the cytoskeleton, mitochondria, lysosome, Golgi apparatus, smooth ER, rough ER, the cell membrane. So we're going to talk about all these guys. And here's a plant cell structure for you to take a look at. Here, chloroplast, Golgi, smooth ER, rough ER. This is the central vacuole filled with water. Plants have mitochondria, people. Don't make a mistake on that. I usually try to test you on that too. So make sure to make a note that plants have mitochondria and chloroplasts. So we're going to go through each organelle of the eukaryotic cell, and then hopefully we'll be done. So nucleus stores genetic information, aka the DNA, in these linear chromosomes. The nucleus is surrounded by what's called a nuclear envelope or nuclear membrane, which again keeps all the DNA in the nucleus. And then they also have, in the chromosomes, DNA is organized with proteins to form what is called chromatin. So these DNA is wrapped around these proteins, kind of condensed basically because there's so much of it. It's wrapped around these proteins, and when it's in that form, it's called chromatin. So here's again a picture of the nucleus. You see nucleus here. This is the nucleolus. So the nucleolus is where ribosomes are made. So you should make a note of that as well. The nucleolus is where ribosomes are made. And then outside the nucleolus, you have DNA wrapped around proteins called chromatin, which is called chromatin. And then you have these nuclear pores, which allow DNAs or RNA to escape out of the nucleus if need be. Ribosomes. Ribosomes are where protein synthesis is, where protein synthesis occurs. So this is where proteins are made. It's composed of proteins and what is called rRNA. Don't really have to worry about this until later. We're going to talk about different types of RNA later. So just make a note that that's kind of what the structure is made out of. And then ribosomes, important note to make. Ribosomes are found within these liquid, which means that they're kind of free floating around the cytoplasm, but they're also attached to internal membranes, which you kind of remember with the rough endoplasmic reticulum. So again, ribosomes can be found floating around the cell, and they can also be found at the rough ER. So speaking of ER, we're going to kind of talk about that, this whole kind of endomembrane system, which includes the ER, which includes the Golgi apparatus as well as the lysosomes. So the endomembrane system is kind of the term that is applied to all of these grouped together. They're kind of a series of membranes throughout the cytoplasm, and what they do is they divide the cell into compartments where various cellular functions can occur. For example, in the rough ER, that's where protein synthesis occurs. Smooth ER, that's where we make lipids. Golgi apparatus is where we ship and package. So again, I know this is new, but it's a name to group all of these organelles together. So we're first going to talk about the rough ER. I already mentioned that rough ER's function primarily is to make proteins because they have a bunch of ribosomes attached to it. Again, so when you look at a picture we'll look at a picture pretty soon. We'll see a bunch of bumps on this rough ER, and those bumps are ribosomes. And a lot of times these proteins will be secreted. They could, if we wanted to digest them or break them down, we could send them to the lysosome, or we could send them to the plasma membrane to leave the cell and go to someplace else in the body. The smooth ER doesn't really have many ribosomes, and usually pictures never show that, but their function primarily is to make lipids. So whenever we need to make fats, whenever we need to make membranes, that's where we make them, is the smooth ER. It's also used for detox. So kind of, if you didn't know, your liver is your primary detox organ. And what I mean by detox is detoxification. Our body is able to break down certain poisons. Primarily, a great example would be ethanol, the alcohol in alcoholic beverages. The ethanol is poisonous to us. So if we have too much, it will kill us. But as we drink it, as a lot of people do, your liver can break down that alcohol so it doesn't become poisonous. But if you have too much in too short of a time, you can die from it very easily. So your liver, my point being is your liver cells are very rich in smooth ER. So here's a picture of rough and smooth. Almost always the rough ER is right outside the nucleus. Almost every picture you'll see, rough ER is right outside of it. And then the smooth ER is usually right outside the rough ER. Golgi apparatus is a flattened stacks of interconnected membranes. Their function is primarily to package and packaging and distribution of materials throughout the cell and also outside the cell. So if we wanted to send something within the cell, we'd use the Golgi. If we want to send something outside the cell, we use the Golgi apparatus. And it's kind of a synthesis of cell wall components as well. So if for a plant, if the plant needed to make a cell wall, we use the Golgi to make the cell wall. So here's a great picture of kind of how the transport of various, particularly proteins, occurs, and you're going to want to pay attention to this because this is a very likely essay question. So information from the nucleus, or information from the DNA, which is in the nucleus, is transferred to the ribosomes of the rough ER. Those ribosomes use that information to make a protein. That protein is then sent off from the rough ER to the Golgi. The Golgi will then package it and into this what's called a vesicle, and that vesicle will then send it out of the cell through the cell membrane. Okay, so being able to describe this way that we are able to transport proteins outside the cell is very important. More than likely you will have an essay on this. So please pay attention to this slide. Lysosomes are membrane-bound vesicles containing digestive enzymes. So this is how we kind of digest materials. You probably remember that from past classes. So on the reverse side, if we wanted to get rid of a material, we do the same thing, and then we send it to the ribosome, or send it to the lysosome, which will then digest it. And so again, using the lysosome. Vacuoles are membrane-bound structures that are used for storage primarily. So they're used for storage. The central vacuole on a plant is very noticeable, so we use that a lot. It's used for H2O storage, water storage. Some protists use what is called a contractile vacuole for movement. They'll actually shoot water out of this vacuole and use it for propulsion. And then within us, humans, we use vacuoles for storage of food and other materials, like glycogen. Mitochondria, their primary function is to make energy, create ATP, cell energy, for the cell. So they're again making ATP, cell energy. And it's found in all types of eukaryotic cells. They're surrounded by two membranes, a smooth outer membrane, and then folded inner membrane layers, which are called cristae. And then the inside of the inner membrane is called the matrix. And then the inner membrane space is located between the two membranes. I'll show you that. And the mitochondria are very unique because they contain their own DNA. So you see that here. You see the outer membrane, very smooth. The folded inner membrane. So again, these folds are called cristae. They have their own DNA contained in the matrix. This whole space right in here, this is all the matrix. And then the inner membrane space is the space between the outer membrane and the inner membrane. Okay, that's it for right now. This is part 1, so we'll finish it tomorrow for homework. Thanks, guys. Talk to you later. Bye.